Great. Well, so thanks for having me here, and thanks, Mike, for the introduction. So I'm here to try and give you an overview of what I've been doing my PhD on. So it's quite a lot of material to cover. I'm not going to go super in-depth. I'm just going to try and pick out some of I think the most exciting things I've learned, the most interesting things. So I'll first give you a bit of background into the Hawksbill turtle and its biology, its legal status and conservation status, and then get into my research questions, which are in 4 main areas. And then finally, if we have time, at least, and for anybody who's particularly interested, we could talk about sea turtle species identification. So I have a lot of photos to help us look at the differences between the species, and we have the shells as well for you all to come and look at. So hawksbills, like all marine turtles, have delayed maturity. They don't mature until they're about 20 to 40 years old, possibly. That's the age at which they're first able to nest. This diagram shows their life cycle, and if we start from the nesting stage, we have the hatchlings now leave the nesting beach. They disperse to open waters, and they have an oceanic stage of perhaps 5 to 7 years long. It seems to be fairly variable. And at this point, they're tiny little hatchlings drifting around the ocean. So the currents are carrying them, and they get dispersed quite widely based on the paths of the ocean currents. At the size of about a dinner plate, say maybe about the size of this binder, is when they make a sudden shift in their lifestyle, going from a pelagic lifestyle, drifting near the surface of the water, to now coming close to shore and diving to feed on seagrass beds, coral reefs, rocky habitat near to shore. So it's a very big change in their habitat, and that's described as the neritic stage. And that's basically where hawksbills will spend the rest of their life, on those kind of near-shore habitats. Once they reach maturity, they may make a shift from one location to another, but it will be a similar habitat that they're moving to. And from there, they tend to nest every 2 to 3 years, and they'll make big migrations across the ocean basin. So what I want to highlight is the fact that the hatchlings get dispersed very widely. So when you have turtles leaving a nesting beach, say from Tobago, our turtles might be dispersed across the region. So this brings up issues for management, because across the region, when you look at the protection of turtles, their legal status in all these different countries, it's very variable. So right now, for example, they're completely protected in Trinidad, but not in, say, Grenada, I believe St. Vincent, Antigua, a lot of the other islands going up. And these are animals that we share with those countries because they're so migratory and pass through all our different international waters. So for example, around Tobago as well, in our foraging habitat, the coral reefs, we're going to have a mixture of turtles that probably mainly not come from Tobago Beach, but they came from beaches across the Caribbean as well. So part of my work examines where our turtles are coming from. Hawksbills are described as keystone species, which means that they have an important role to play in the environment, a unique role that cannot really be filled by any other species, and that's because of their diet on sponge. So sponge is a invertebrate, grows on a coral reef, and the top picture you have on the left-hand side a coral animal, and on the right a sponge, and they're actually literally competing for space. And sponge often outgrows coral and can grow over coral. So it can end up changing the structure of the reef environment. If you take out the hawksbill turtle, which is such a large animal that will feed on a lot of sponge. Sponge is its primary diet, and you don't really find that among other reef species. You have a few other fish that feed on sponge, but not in the volumes that hawksbills do. So they help maintain biodiversity on reefs. Now, when I first started this project, it was 2007, when it was still a legal open fishing season. So that was one of the things I wanted to examine. So at the time, these were the rules. All turtles were protected for 7 months of the year, between March and September. You couldn't touch any turtles. But outside of that, you could hunt turtles offshore. There were certain rules about females in particular were being protected if they were on a reef or 1,000 yards from shore where there is no reef, which was very kind of cumbersome and difficult to enforce. Now, since 2011, this has completely changed and sea turtles are completely protected. And that's a Hawksbill turtle that's being sold at a fish market in Tobago. Here we have green turtles in wild Waiari. Before I started my project, all you can find about turtle fishing locally is that it's a small artisanal fishing industry focused on greens and hawksbills, which are the most common turtles we have offshore. But the harvest was unregulated and not monitored. So any details of any records of how many turtles are being captured or the weights of the turtles is very hard to find, and it's definitely incomplete data set. So that's why I wanted to examine turtle fishing as well. So apart from this active and unregulated turtle fishery, and you have people claim turtle meat is a part of our culture. It's things that meat especially used on typically for harvests and events like that in Tobago, not a day-to-day thing, but special events. On the other side of things, Tobago has a growing dive industry. The map shows the extent of the coral reefs around Tobago, although it's even more than that really. And the World Resources Institute has shown that coral reef associated tourism is very valuable to us here. So with that in mind, my main questions of interest were to learn about the local aggregation of turtles we have on our reefs around Tobago. Then I wanted to look at what rookeries do our foraging turtles come from. So what nesting beaches are these turtles coming from? And now I'm also looking at where do our hatchlings on Tobago leave and end up. I looked at the extent of the local fishery, and then I'm also trying to look at the potential value of turtles to the growing dive industry. So between 2007 and 2008, I conducted an in-water survey using scuba gear. So I conducted visual swim surveys underwater, and I did a market capture on turtles. Here we actually have a green turtle. This is on Mount Irving Reef, and you'll see I actually found a lot of green turtles there. So this is me with various turtles, just showing you how I capture them by hand, bring them up to the boat, take measurements from them, tag them with unique identifying tags, and release them. So what I really wanted to look at was just to measure relative abundance, to see were turtles more abundant in any one location. So is there any special habitat that we should be particularly concerned about protecting? Usually measuring relative abundance, measuring how many turtles you see for the length of time or for the distance you walk is a good method. But there are severe limitations with my study. One, because you're underwater, scuba diving. It's difficult to accurately measure what distance you traveled. You've got currents of varying speeds, you're not going in a straight line, you can't use the GPS on the water. So turtles per unit time as well turned out to be difficult because of my method where I was capturing turtles. So it actually found that when I capture a turtle, I end the dive. So that shortened the dives, and that would affect my data. So the main factor I'm looking at, the main estimate of relative abundance, which eliminates that problem of the shortened dives, is the frequency of turtle encounters on dives. So overall, I did 235 dives, that's almost 190 hours underwater, and we had 130 turtle encounters, which gives us a rate of either 0.55 turtles per dive or 0.69 turtles per hour, depending how you measure it. Overall, we found turtles on 40% of all the dives I did. And out of all the sites I did, I studied, I think it was 43 sites I visited, and we had turtles encountered on 31 sites. Here I just have side-by-side hawksbills and greens, so I did do some work on green turtles, but because they're not as abundant as hawksbills, my main study is on the hawksbill. So you have, compared to 111 encounters with hawksbills, only 19 with greens. And of course that gives us very different numbers for their encounter rate. And turtles were more limited to a handful of sites. Only 9 sites we encountered greens, and 16 out of all, 193 dives. Sorry, 235 dives. This map gives us an overview. This is sticking with turtles per hour, so the unit of time is a measure. We have hawksbills in blue, and it's supposed to be kind of red, but it's showing up black. So I want to highlight a couple areas, basically. So down here off the southwest of Tobago, this site was particularly important for hawksbills. I really found a high abundance of turtles there. Only a few short dives, but every dive we saw several turtles. And that rate was about over 8 turtles per hour. The other site I want to highlight is this area of Mount Irvin, which was the best area for green turtles. So green turtles were found in Speyside and down in the southwest, but Mount Irvin, they were really abundant. And actually at one of the sites, they were even more abundant than hawksbill turtles, which was interesting. A lot of people associate green turtles with only seagrass beds, but they're found on coral reefs too because they feed on algae. So my effort at the dive sites varied a lot, by the way. So I was doing this study with the help of dive instructors in Tobago, dive operators who have their own boats and are taking their tourists out. So I would just go wherever they went. So of course they frequented some sites more than the others. And that's a limitation in the analysis of my data as well. Here we have all the dive sites separated by habitat type. I'm sorry, the colors aren't showing up too good. But the blue is what's most important when you look at the analysis. The blue sites are the sites that were dominated by sponge rather than coral. And recalling that sponge is a favorite food of hawksbills, it makes sense that when you looked at the frequency, the proportion of dives with turtles, sponge sites had much more abundant turtles, hawksbill turtles that is. So when I compared sponge with all the other sites, I found that hawksbills were actually more abundant wherever you had habitat dominated by sponge. Alright, this is this is the same data, but looking at exactly how many turtles I saw on how many of the dives. So you can see the shape of the curve is different. So coral and rock is a very steep curve because a large proportion of the dives we saw 0 turtles. However, you've got the sponge here. It's not as steep because there were so many times that we actually saw hawksbills, and the statistics support that conclusion that hawksbills are more abundant where you have sponge. So this graph now shows the turtle sizes we saw. So this is important just to get an idea. Is it a lot of adults we have? Is it juveniles? And here we can see for comparison, the hawksbill size at maturity and the green size at maturity. So we did have a number of adults in the water, but the majority were subadults and juveniles. And this is data based on all the encounters I had and estimating their size by site. I only was able to capture about 50 hawksbills and 6 greens. And that would have been biased towards size as well, because the bigger the turtle, the harder they are to catch. So that's why I wanted to look at the entire data set. So I'm just going to highlight here, when I looked at size, and I compared it with habitat type to see if there was any difference in size according to habitat type, but I also looked at the regions. So I split up all the dive sites into 3 regions around Tobago: the West Atlantic, the Caribbean, and Speyside areas. And when you split the graphs up like this, you can just about start to see it here that the Speyside area in yellowy green, the curve is more shifted to the right. So we actually had larger turtles found in Speyside than elsewhere. Okay, so there's this is the size the turtles first usually recruit to the population, and they were very abundant in the Caribbean and West Atlantic, but virtually absent in Speyside. And then you have this middle group here of subadults. Very few in the other areas and many in Speyside. So that was an interesting result. And my hypothesis to explain this, and this of course deserves further work, is perhaps hawksbills are actually recruiting or arriving into Tobago only on the Caribbean and West Atlantic regions. And then as they settle in and they grow, they're making a shift in their habitat from one area into Speyside. So it's a thought, and it may have something to do with the strength and direction of the currents as it passes around Tobago. The currents in Speyside are very strong. So perhaps the young hawksbills are better able to make the transition in the Caribbean and West Atlantic regions. So the next part of my study, so while I was tagging these turtles, the value I gained from tagging them was the ability to look at genetic analysis, because I had identified individual turtles and I knew I could get samples from each individual without making the mistake of taking repeated samples. So I want to introduce you all to mitochondrial DNA. This is DNA that's found in your cells in a special organelle called the mitochondria. It's a circular piece of DNA, and what happens, I'll show you here. If you look at a family tree, you know with regular DNA, your nuclear DNA, you get a combination, a mixture of DNA coming from your mother and your father. Mitochondrial DNA is different. It comes as a discrete package, no combination, no mixing up, from the mother to all her offspring. So from the mother to her daughters and sons. So using mitochondrial DNA, we can trace this maternal lineage back. And just to get you familiar with terminology I'm going to be using. When we're looking at DNA, we talk about haplotypes. A haplotype is just one sequence, one portion of DNA that we look at, a particular segment of the DNA strand, and it forms a unique sequence or pattern. So it's these four letters, A, T, C, and G, which represent the bases on DNA, and they all are mixed up in different patterns. And when you look at individuals, you can compare them against one another. So this strand here is just 400, not even 400 base pairs long. And I was looking at strands almost 800 base pairs long. And just one change in one letter creates a new unique haplotype. So that's the kind of data I'm working with from that. And these are types of studies that have been done around the Caribbean already. This map is the different colors are about 20 different haplotypes that have been identified at rookeries around the Caribbean, or nesting beaches around the Caribbean. So these are the various nesting beaches where these studies have been done. The size of the pie chart gives you an idea of the size of the population, and the colors show you the different haplotypes. So I want to point out there's usually not, there's a distinction, not that you get only one haplotype in each beach, but it's the mixture, the unique mixture of which haplotypes you have that's important. So it's the relative frequency of haplotypes we're looking at. And this data also demonstrates the idea of natal homing, which is where females always return to the nesting beach of their birth. So by the females always going back, you're collecting their mutations in that population, and you can see distinct populations when you look at it genetically, based on the frequency of haplotypes. And looking at the rookeries and then looking at it with our foraging turtles, what we can do is a comparison to estimate where our turtles are coming from. So part of the process was just taking a small piece of tissue off the edge of the flipper using a biopsy punch. And that's what we work with to get the haplotypes. What we found was 6 haplotypes were present in Tobago, and that's represented by these bars here. So you can see that 2 of them are the most common haplotypes in the whole Caribbean region. These haplotypes are present in the majority of rookeries and in large proportions. But a couple of the others are interesting. This one has only ever been documented in Costa Rica, in a rookery in Costa Rica. It would have been documented in other foraging grounds. This one is found mainly in Mexico. This one's never been documented in a rookery before in foraging grounds, so you don't know where those ones are coming from, but it's been found in other foraging grounds. And likewise, this one has been only found in Brazil. So looking at it that way, it gives us an idea where we can pretty much say we definitely getting turtles from Costa Rica, Mexico, and Brazil. But we want to know more. We want to know an estimate of proportions, how many are coming from each different area. So the statistics I use is called Bayesian mixed stock analysis. The idea is you're using prior information, including the size of the population at the nesting site, together with the unique haplotype frequencies you get there, and you're plugging it into the program along with the foraging grounds that have been analyzed. So up to date, there's been 10 nesting grounds that have been characterized, and there's 11 foraging grounds that have been characterized before Tobago. So we add Tobago into the picture, we've got 10 rookeries, 12 foraging grounds, and it even can estimate for there being other unknown foraging grounds. This is a peek at the data. So these are the different source rookeries where our turtles are coming from, and the proportion that it contributes, the proportion that makes up Tobago. So we're taking Tobago, say our population, 100%, 35% is coming from the leeward coast of Barbados. Another interesting thing, this is the windward coast of Barbados. Those two populations, though they're so close together on the same island, are distinct genetically. And that's been shown by previous work. So it's very interesting, a large proportion coming from Barbados, and after that Brazil. But we're drawing a small number of turtles from all across the region. The statistics also allows us to look at it from the other angle. So that was looking at it from the Tobago point of view. Now if we look at the point of view of Barbados, although their turtles make up 35% of our population, the fraction that's coming to us is only 12% of their population because their population is so large. So this helps us look at, for example, a fishery in Tobago. How are we impacting around the region? And you can see when you look at it just from the Tobago point of view, it looks a lot worse, that we're probably impacting Barbados a lot. But when you look at it from their point of view, it's a smaller fraction of their population. But these are still significant numbers. So this map just puts the two together, I hope, for your understanding. So again, the proportion of these pie charts shows the relative size of the population. The fraction of pie that's missing is how much of their turtles come to Tobago. And then when you're in Tobago, this is the proportion that it makes up. So again, for example, Barbados, 12% about of their population forms 35% of the Tobago foraging aggregation. Together, Barbados and Brazil make up more than 50% of our Tobago foraging grounds. Right, so this, every time someone does a DNA study like this and adds another foraging ground, another nesting ground into the study, it helps us better figure out regionally where these turtles are moving to and figure out the relationships between the nesting grounds and the foraging grounds. We found the aggregation here is a typical what you call mixed stock derived from all over the region, very high diversity. And Barbados and Brazil contribute the most to our population here. So this is where I'll just mention that was the first part of my study when I was doing the master's. What I'm doing now is trying to characterize the Tobago rookery, the Tobago nesting turtles. So SOS Tobago and a group called Nest in Tobago also helped me collect samples now from the nesting turtles, the nesting hawksbills, and I'm about to start analysis on that to repeat the mixed stock analysis or the statistics incorporating the Tobago rookery. So that will further clarify what we know and help us see if, what proportion of our own turtles actually stay here around Tobago. Furthermore, we can look at the relationships among Tobago and the other regional rookeries. And this is what's been elucidated so far from other studies around the region. And you can again see strange relationships with Barbados Leeward and Windward, while they're so geographically close to each other, are very genetically distinct. They're more closely related to these groups of countries. So all of this is still, it's like putting together puzzle pieces to try and figure out the relationships. Alright, so the turtle fishery, I'll try and run through this fairly quickly. I got the help of locals in Tobago to help interview fishermen. There's about 800 registered fishermen in Tobago. We surveyed 215 fishermen distributed around the island, roughly according to which beaches are more active in terms of fishing. So, for example, Charlottesville is one of the most active areas, so we did a high number of surveys there. So out of those 215, we found 22 fishers who would admit freely to turtle fishing. And you can see that they were found all over the island. And that's an important thing because before, if you read a lot of documents about turtles in Tobago, they describe the main area for turtle fishing as Kilgoin, Crown Point, Pigeon Point area. But it's for sure all around the island. So here I just have some statistics to highlight some differences I found between those who fish turtles and the other fishermen. So while their age was the same, I found that turtle fishers had actually spent more years fishing, which meant they had started fishing at an earlier age, further back in the past. Maybe this says that it's kind of a dying tradition. Perhaps it's the fishermen who started up younger, earlier, were encouraged to fish in turtles, but it's losing that appeal. Registration, I really don't know why, but we found that a significant number of turtle fishers they tended to be less likely to have registered with the fisheries division. The registration with the fisheries division doesn't really, it's actually not legally required per se. It means that they will get tax deductions and help with their boats and their equipment and things like that. So we're not sure why this difference. Education was interesting because turtle fishers tended to be less well educated or had completed less stages of school. We also asked all the fishermen about their opinions and knowledge about the laws in Trinidad, the origin of Tobago turtles, and the abundance, and what they think about abundance in Tobago. So it was interesting that half of turtle fishers said, oh yeah, there's plenty of turtles now. Only 12% of all the other fishers said that they were abundant. They wouldn't use abundant to describe them, they used other words of less, fewer turtles. And when we asked them also about the change, has there been a change? Do you think there's fewer turtles now than before? Almost half the turtle fishers said there's fewer now, but almost 90% of all the others said yes, there are fewer turtles now. So perhaps the turtle fishers were trying to, play down the loss of turtles, the low abundance of turtles, and claim that, yeah man, there still are plenty, we could still go and fish. So when we talked to fishers about their effort, many of them, 5 of them didn't give us any details at all about how often they fish. Of the others, 5 said a few days a year, 5 said a few days a week only in the open season. 1 said he does daily in the open season, and 6 said they fish daily year-round. After that, we also asked, well, what's your annual take with that effort? Again, a lot of them wouldn't give us an answer, but when you look at the numbers, this 120, 18 visits, 150, 6 to 20. So that's just from 9 who reported on their take. Those 9 fishermen together catch probably about 370 turtles. That's ignoring 13 fishermen we don't know about because they didn't give us any data. But the 13 includes the 6 with the highest effort, those who fish daily, year-round. So the 370 is definitely an underestimate. It's difficult to extrapolate based on this. And again, our survey didn't cover all the fishermen in Tobago. The survey covered maybe 20% of the fishers in Tobago. So when you extrapolate these numbers, it gets into tricky territory, but it's probably quite a significant number of fishers. And again, this data was from 2007. So we wanted to look at the income. Turtle meat was generally higher priced than regular fish. $25 to $30 per pound is most often what they said. Only 14 out of the 22 said they consider turtle fishing an important source of income. And that varied from 13 to 40% of their entire income from fishing. And when we looked only at those fishers whose main source of income was fishing, there was actually no difference in the annual income. So it shows that turtle fishing didn't improve their income. So basically, turtle fishing is happening all around Tobago, year-round by a lot of fishermen. And this was while there was an open and closed season, but they freely admitted, a lot of them, that they fish year-round. A small number of fishers are having quite a significant take, so it's lets you know that what a lot of fishers might be doing together. Enforcement is clearly inadequate because they, were happy to go around fishing year-round. A lot of them didn't even know or understand the regulations at the time. Most of them thought that turtles in their waters are Tobago turtles. They don't understand that they move around so much and that they might be impacting turtles from other destinations. And monitoring of the resource and fishery, I confirmed, is really non-existent. So there's some Hawksbill meat on sale in Tobago again. And basically, the work I've done, I think, really supports the decision of the government to finally close the season in 2011. For numerous reasons that needed to be done. There's no way we could claim it's sustainable if we don't have an estimate of what is there and what's being taken. And neither of those have been met. On the other hand, because it seems to be a relatively small number of fishers doing this activity in turtles, I think that's hopeful that it could be addressed. Income doesn't seem to be a great incentive to continue fishing turtles, but there's certainly always a cultural thing that they feel this is their right, this is what their parents, grandparents did, they should have the right to be able to continue. And from the data on the education and the age of the turtles, the activity of them over the years, perhaps with improved education and over time, turtle fishing might be falling out of popularity. So, economic value of turtles. I wanted to look at the alternative now. We looked at the value of consuming turtle meat, what the meat is valued, but what about live turtles that visitors see while they're here? Now, hawksbills you don't usually see on the nesting beach very much, so I wanted to concentrate on encounters in the water, in particular while diving. So it's not something that divers pay just to go and see a turtle. The turtle is just like a highlight on an addition on their dive that they're doing. So when we talk about value of turtles, there's all these different ways you can look at the value of turtles, and I'm focusing on the ecotourism aspect. Right, so like I said, turtles are not, it's not like a nesting beach activity where you pay to go and see turtles. You're paying for a dive, but if you see a turtle, it's an added bonus. The image has been used quite often in Tobago advertising, and because they're not the good that people are going out to buy, like the nesting turtles, you can describe as a market good, because people are directly paying for that experience. Here they are non-market good, just a part or component of the dive. So I use a method called choice experiments, where you're studying stated behavior in a hypothetical setting. So you're going out and talking to people and you're saying, if I give you option A and option B, what will you choose? And based on those choices, we can figure out what they prefer to see on dives, and how much they would like to pay for those dives. So what we do is we have two profiles in my design describing a dive. And I'll show you the different attributes, but of course turtles and price was the main thing I was interested in. And each profile in the choice set has different levels of the attribute, and respondents end up having to make trade-offs. So there's pros and cons to each option they're presented with. And based on their personal preferences, they'll make the choice. So these were the different attributes we looked at: price, turtle encounters, but also coral cover, fish diversity, and the crowding, because divers don't like to be too crowded when they go out on a dive. So these were the figures we used to plug into the choices. And here you have an example of a choice, right? So option A on the left-hand side, 100 US, it's more expensive, but you're getting more fish species, fewer divers, only 5 other divers, and you're still seeing a turtle. On the other hand, the coral cover is very low. It's not the best looking, healthiest reef. Option B now, it's cheaper, and it has a much healthier reef, but you're seeing fewer fish species, and you're more crowded. You've got more divers around you, disturbing the fish and so on. But you, on the other hand, also got 3 turtles. So these are the types of choices that the respondents get to look at. And each person has to look at 6 of these. Overall, there were 96 that are presented to the whole sample. And just an overview, this data is still being analyzed, but what we've already discovered so far is this is the willingness to pay for a diver to see these different conditions. So, for example, coral cover, divers are only willing to pay 9 US for the low quality, 36 US for the medium, 95 for the best coral cover. Contrast that with turtle encounters, which starts at 93 US. So turtle encounters of these 4 factors was actually the greatest driving force to pay money for a dive. They're willing to put out a lot more money just for the guarantee of 1 turtle encounter when you compare it to coral cover and fish diversity and so on. And of course, the value goes up, but with what you call diminishing marginal utility for any econ people. But it starts high and just small increments up as you see more and more turtles. Right, so I mentioned already that I have a further mitochondrial DNA work of Tobago, Hawksbill, rookery, and recalculating the MSA. And in addition to the economic work, I'm doing another survey of tourists to examine their willingness to pay for conservation. So this is not how much they're willing to pay to actually see a turtle in the water and so on, but just hypothetically, would you be interested in making a contribution to turtle conservation, and how much? And I'm going to be looking at, how the experience influences that. So I've been interviewing people who've been diving, snorkeling, have seen turtles nest or hatch on the beach. And it's already clear that, if you've just seen baby turtles hatching on the beach, you're hooked and you want to give money. So it's another estimation of their value that I want to look at. So overall, through all the areas of my work, we're contributing to the regional picture of Hawksbill migration, a new level of understanding of the foraging turtles here. I mean, no one has done a study like this before. We had no idea what the state of the population was like. So with the information gathered, we can now establish a regular monitoring program, select critical habitat for protection, and look at implementing education of fishers and enforcing regulations. Other future things that I think definitely should be done is look more closely at why the variation in turtle abundance around with the different habitat, and why the difference in size around the different regions. There's great potential, I think, to get the local dive operators involved to actually collect this monitoring type data. You don't have to catch turtles to monitor them, you just have to record how many you see when you're on a dive. So dive operators are the ones who are in the water all the time, and my next hope is to enlist them to start recording this kind of data. But the addition of tags in a more long-term monitoring program as well will help us understand more about how the turtles are using the habitat and moving around Tobago. So for example, that idea of maybe they're recruiting in one area and shifting around to Speyside as they grow. One way we could examine that is the use of tags. So that's the end of my presentation on the PhD aspect of all my work. And we can probably stop here for questions, and if there's interest in looking at the identification of turtles, I have a lot more pictures of the different species that we could examine as well, and you're all welcome to come in.
Okay, thank you very much, Michelle. Right, now because we've got this extra little session potentially looking at species here, I'd like to keep the questions short. Okay, focused and short. So a couple of questions, and then we'll get on to having a look at the species. Okay.
In the pelagic mode, what do they feed on?
Good question. Mainly algae at the surface. So a lot of loggerheads, for example, we know have been tracked through the Sargasso Sea, and they're closely associated with the Sargasso algae drifting around, and they're using that for protection as well as food. So there's a diet shift. We think the hawksbills do similar. And so along with the habitat shift, there's a very big diet shift from vegetation to sponge.
If I could complete questions. Sorry. There's something I didn't really understand with respect to the statistics.
Okay.
Is it that when you examine the DNA, in many cases you're getting unknown? Because since you're getting samples of, say, Brazil, two sides of Barbados, but you're not seeing anything from St. Vincent, St. Lucia, and so on. Is it that when you examine it, there are unknowns, that you're getting turtles that you don't know where their DNA is coming from?
Yes, basically. So first of all, there's a limitation in the available data. So no one's done a study in St. Vincent, St. Lucia, and all of that. So we can only work with what we have. And then if we looked at the graph with the haplotypes, so what I was saying is, you can't tell where an individual came from necessarily. If you go back again to the graph.
Okay, I understand.
Yeah. So it's a matter of looking at the haplotype frequencies will give you an estimate of the proportion, but you can't really, so in most cases you cannot say this turtle came from Brazil. Yeah.
Since the change in legislation exactly 2 years ago, have you noticed any or is it declining rate of poaching?
I have not been back in the water as much, or back in Tobago as much as I would have liked since then, so it's very hard to say, but I don't see any reason why. Because if you, for example, the previous laws with the open and closed seasons were around since 1975, and nothing happened, like, they don't, it didn't make a difference really. So now this further change, I'm not sure if the fishermen are even aware of it, and I'm pretty sure most of them don't care. I can't actually speak about nesting poaching because last year summer, I spent a few nights on Buku Beach in Tobago, just 3 nights. Every night we found a hawksbill, every night it was in the process of being poached. So it's certainly still a major problem in Tobago. In Trinidad, we've been able to manage the leatherback nesting beaches because all the leatherbacks are concentrated on small areas, but in Tobago, the hawksbills in particular nest everywhere on lots of secluded beaches, and we just don't have the manpower to patrol and enforce. So I mean, that's an example. That was last year after they closed, and people are still hunting nesting turtles, and nesting turtles have been illegal to hunt since '75. So there's a very long lag in the law coming in and actually having an effect, because the enforcement is not there.
Are you saying that the fishermen catch the turtles on the beach, or do they catch them at sea?
Okay, the fishermen are generally catching them at sea. Yes, they set nets, they use harpoons or spear guns, and they capture them at sea. And they know they have special gill nets that they like to use right on top of a reef or on top of a seagrass habitat, where you usually wouldn't catch any of their other target species. So it's the turtles they're going after, and they describe, they'll leave the net there a few hours in a day or even overnight and come back to check it and stuff like that. So the poaching on the beach is probably a different group of people, or there may be some overlap. But
So
what I was examining was more the fishing, catching in water.
You can't exonerate them by saying they're accidental capture.
No. And they said, yes, I catch those. There were a few, I think maybe 40 out of the 200 who said they accidentally catch them from time to time. But what I was focusing on is those who target.
Okay, there's no more questions. We'll just put some special reference bench and then.
So we could go. I have some more pictures, and we could look at like species by species if you like, basically. Right, so there are 5 turtle species, the leatherback, and these 4 hard-shell species. So I think everybody's pretty familiar with the leatherback, and we'll focus on the other 4. When identifying hard-shell species, a lot of people, I find, focus too much on things like coloration, the shape of the shell, whether they have overlapping scoots and things like that. But the primary things we need to look at are the pattern of the scoots, which is like the scales on the back of the shell, the carapace, and the number and pattern of scales on the head. Those are the only defining characteristics, and it actually makes turtle ID very easy. So basically, often what we need to do is count these number of scoots on the side. These are the costal scoots. The number can tell us what species, or at least narrow it down. The prefrontal scales are the scales between the eyes, and that again can tell us either for sure what species, or at least narrow it down. And the combination of the two helps. Then also we look at the position of this scoot to this one. It can either be separate like this, where it doesn't touch, or this one could be jammed all the way up here and actually come into contact with the nuclear scoot. These are the prefrontal scales, so it's just those between the eyes you're looking at, and that's an example of a Hawksbill. The hard-shell turtles have claws on their flippers, and they can either have 1 or 2 claws, so counting the number of claws can also help you add up the data. So overall, green and Hawksbills both have 4 costal scoots. Loggerheads and olive ridleys have 5 for loggerhead usually, and 5 for loggerhead usually, and 6 or more for the olive ridley. So whenever you have more than 4, basically, that means that the costal scoot is going to be touching the nuclear scoot. So for example, I've been able to identify pictures of turtles that only show this region, right, near the head of the turtle, because you can see where this position of the costal scoot is. You may not be able to count, but you can see the position and say at least whether it's the top 2 green and Hawksbill or bottom 2 loggerhead and olive ridley. The prefrontal scales, green is the one that's unique, only a single pair. So as soon as you see a green head, you're supposed to know that's a green turtle. The others gets a bit more complicated. So you can get keys like this online that help you figure it out step by step. Basically, if it has a bony shell with scoots, then you're going to count the number of costal scoots. Does it have 4 pairs or 5 or more? If it's 4 pairs, it has to be a Hawksbill or green, and the deciding factor is whether it has 1 pair or 2 pairs of prefrontals. If it had more than 4 costal scoots, then you count them again, basically, and 5 is generally a loggerhead, and 6 or more olive ridley. So the Hawksbill, we just look at the various pictures of them. So the shape of the beak is also usually stands out in a Hawksbill, but not everybody can recognize the difference when you have a Hawksbill next to another species. But you can see how the 4 scoots makes like the shape of a cross between the eyes. And they always have 4. It's set like that, whereas some of the other species are more variable. People always say, well, hawksbills have overlapping scales, but they don't always. Sometimes they have a very nice smooth shell and no sign of the overlapping. Overlapping tends to be more in the juveniles, but as they grow, they leave that behind, basically. Some pictures of the hatchlings I have, you'll see that 3 of the species are very close in color, shape, size, and the only way to tell is going to be counting these costal scoots. The green turtle. People don't realize the variety of colors that the green turtle shell can be. You can get very light pale ones, very, very dark ones, mottled ones that look almost like a hawksbill. So the color of the shell is not the best factor. But the 2 scales on the front of the head is really what you want to look for in a green turtle. No other turtle has that.
Question. Why is it called green turtle?
The story is because the color of their fat is actually green. When you dissect a turtle and look at it in its meat, it has a layer of very dark green fat. So it's nothing to do with the external color, which a lot of people assume. And again, the variation in color here, but the consistent thing is the prefrontal scales. Now, who can tell the difference between these 2 then? Right, so this one is the hawksbill, where you can see the cross clearly in the hawk-shaped beak. This one, the colors are very similar, but it's a green, and you can confirm by the prefrontal scale. So just put that in to emphasize how similar the shells can look, and even the color of the scales and so on. The green hatchling is very distinctive. It's very dark with this bright white line around the edge of its shell. Yeah, that's a green hatchling. So the loggerhead is named for the size of its head. It feeds on shellfish and like conch and lobsters and so on, so it has very strong jaws for crushing. It sometimes has these ridges, especially when it's younger, down the middle of its back like that, but not as it gets older. So you can see the difference here from a hawksbill, the size of the neck and the head, and there's no sharp beak coming out. And you can also quickly look and see that the costal scoot is touching the nuchal scoot. And you guys can see that when you come and more closely examine these things. So this is a female adult loggerhead. You can see the dome now of the carapace completely. No, this is actually, I don't know it, of the internet. For example, I am yet to see a loggerhead in Trinidad or Tobago, but I've seen pictures of at least 2, one in Tobago, one in Trinidad.
In the first publication by Dr. Dilemma myself, the little book, Returns to Trinidad and Tobago, there is a picture in that of a loggerhead. I thought it was green at the time, but check it out. It's a loggerhead. And that was taken in Port of Market.
Okay. Yeah, the 2 pictures I've seen were in Charlottesville, Tobago on a dive, and on the east coast of Trinidad, I think Melandra type area, just a couple of years ago.
I think also years ago, and I'm talking about when the field naturalists used to go every Friday night to Las Cuevas, we saw one and we didn't know what it was, and we took a photograph, and I think afterwards it was identified as a loggerhead on Las Cuevas.
Yeah.
But this is many, many years ago.
Yeah, I'd love to see one in the water.
All those guys and you haven't seen one as yet?
No, and I only know one dive operator who has, for sure at least. But they're not necessarily the best at identifying either. There was a picture she took in Charlottesville, and she said, "Look at this funny looking hawksbill," and I said, "No, it's not a hawksbill, it's a loggerhead." So they do exist, and we did have an olive ridley wash up in Tobago a couple of years ago on the south coast by Lampo. So this is the loggerhead hatchling. You see from the coloration and the size, very close to the hawksbill I showed you before. And the only way to tell the difference is the scoots. Well, it's trickier with these 2 because the loggerhead can have 4, just like the hawksbill. And when they're small like this, the beak isn't as pronounced. So for hatchlings especially, the scoots on the carapace are really important to to look at. And it will tell you right away, you know, so no horrors in looking at the other things. The olive ridley is very distinct because of how many coastal scutes it has and the shape of them. They're very squashed together, so long and thin, and you get at least 6 of them packed together like that. And actually, well, this picture was one in Tobago that had washed up in Lambo with some injuries, and we tried to take care of it. And then it went missing.
But this is the size, is it more the size of a Hawksbill, right?
The olive ridley?
Yes.
It's smaller. Yeah, the olive ridley is pretty much the smallest one we get.
So you shouldn't be typical ones, you can see a young one there.
Well, yeah, so it can be hard to tell when if it's a small one, it could be anything. If it's a huge one, well, green is the biggest, so it's probably green. Yeah. So again, the 3 loggerhead, Hawksbill, and olive ridley next to each other. I mean, these 2 in particular, based on the color, are almost, you know, identical. What you need to look at is, once you look at the scutes, you can see the difference. This little one, this coastal scoot is pushed up, touching the little scoot, and 1, 2, 3, 4. You can't really because of the sand, but that is only 1 here, so it's at least 6.